Iron in ferrous form. This is the new trick. There are two dominant forms of the iron is used, ferrous form and the ferric form. The advantage of the ferrous versus the ferric formulation I will tell you. Okay, so what is the advantage? That the slow release ferrous sulfate preparation remain the established standard treatment of iron deficiency. So whenever there is a iron deficiency, slow release ferrous sulfate preparation remain the established standard treatment. Now the ferrous sulfate do very low or low GIT side effects. First thing. Second, ferrous sulfate has very high oral bioavailability and efficiency in liquid form. It tastes good, so it is acceptable. It is useful in iron deficiency anemias, which is associated with autoimmune gastritis, Helicobacter pylori gastritis. So whenever there is an autoimmune gastritis or Helicobacter pylori gastritis, it is useful. When ferrous sulfate not tolerated, ferrous glycine sulfate or ferroglycine sulfate may be given. Iron is better absorbed when there is a low pH. So the 10% of the dietary or the oral iron mainly absorbed in the duodenum, and in stomach ferrous form oxidized to ferric form. So whenever there is a presence of reducing agent like the ascorbic acid, it will absorb the absorption of converting ferric into ferrous. So we prescribed iron with vitamin C. Stored in the form of iron is stored in the form of ferritin, hemosiderin, myoglobulin, heme protein, cytochrome and catalase. So what is the disadvantage of non-heme iron? Because it decreases absorption of non-heme iron when it is formed insoluble complex with the drugs, with tannin and after meal. Now what is a storage form of iron? It is a ferritin. So the ferritin storage pool replenishes whenever the plasma iron concentration low. So whenever there is a plasma iron concentration low, there is a storage that is in the form of ferritin replenishes it, and ferritin storage pool replenishes it and transported in the form of transferritin. Now what is the unique characteristics of the transferrin? It binds strongly with the receptor in the cell membrane of erythroblast, which is present in the bone marrow. So this is an immature RBCs, that is erythroblast, the receptor which is present in the erythroblast, it strongly binds. So transferrin strongly binds with the receptor which is present in the cell membrane of erythroblast. And these is erythroblast, they are present in bone marrow. So iron inserted or transferrin inserted into the erythroblast by endocytosis process. So by endocytosis, the transferrin allow to enter into the erythroblastin and deliver inside the erythroblast. It delivers the iron directly into the mitochondria where the heme is synthesized. So the deficiency of the transferrin also causes severe hypochromic anemia. Now the absorption of the iron from the GIT, it is how it is absorbed. I would like to tell you how the iron is absorbed from the GIT. It is apotransferrin in the bile which is secreted from the liver. So liver से कैसे secrete होता है? It is in the form of apotransferrin. Apotransferrin in the duodenum, it binds and they form the transferrin. So apotransferrin is converted into transferrin. Duodenum, where the apotransferrin binds with the free iron and form the transferrin. So the apotransferrin plus iron means it is transferrin. Now the transferrin by pinocytosis absorbed in the intestinal epithelial lining after binding with the receptors. So iron from the transferrin later released into the blood capillaries beneath the cells and circulate as plasma transferrin. So what is transferrin? Means it is a protein apotransferrin plus iron. Apotransferrin secreted from the liver. So the liver functions should be proper to do not have the anemias.